Good evening. Good soggy evening. So last week, the folks that were here in the Jackson Center had the benefit because they got the extra cake. This week, I think the folks watching from home might actually have the advantage because they're dry and we're not. But I'm really glad to see you here, especially so many of you braving a cold and rainy night. So thank you for being here. We're in our third week of this year's Biotech 201 as we talk about secrets of the human microbiome. We have so much ground to cover tonight. But having said that, we're going to actually start by taking some of the questions that you have asked me over the last several weeks that I said I don't know the answer to. So I did some research and some digging into them. So one of the first questions that we got last week was about lectins. And specifically, are lectins bad? And this was a question that came up in the Jackson Center, but we also had several folks online that were asking the same sort of question. So let's dig a little bit into lectins. Lectins are proteins that are found in lots of organisms, but especially in plants. And these proteins specifically bind to sugars. They bind to carbohydrates. And they are part of the plant defense mechanism. So throughout history, if you're a plant and something is chewing on you, you have very few options to defend yourself. You can't pull up your roots and run away. So you have to figure out how to defend yourself in place. And often plants have evolved to do that by producing molecules that either don't taste very good or actually are damaging to the insects or the other animals that may be eating them. Lectins fall in that category. So lectins in high concentrations actually stick to the cells in the intestines of insects, for example, or other animals that are eating them. Now, again, the key phrase here is at high concentrations. Now, let me give you an example. Red kidney beans. I would imagine everyone in this audience knows that you should not eat raw kidney beans. Because if you eat raw kidney beans, uncooked kidney beans, you will have the most miserable evening. You may find yourself in the emergency room with incredible intestinal issues, including bleeding and diarrhea and all sorts of other issues. And that's because kidney beans have an unusually high concentration of lectins. Cooking them totally inactivates them. So you can eat cooked kidney beans all you want. The specific lectin in the red kidney beans is called phytohemagglutinin. Now, having said that, lots of vegetables have lectins in them. Most of them, at almost, unless you're eating bushels full, the lectins are not going to give you problems. Now, this is brought up because there are a series of popular diets and books right now that talk about how you need to try to get a lectin-free diet. That if you can cut all lectins out of your diets, all grains, all beans, all carbohydrates, if you can cut that out of your diet, that's actually going to be incredibly helpful for you. And some of the claims are pretty spectacular. Treats autoimmune diseases, cures and prevents cancer, treats diabetes. I think that there are certainly people in this world that have dietary sensitivities and dietary allergies. That certainly is true for individuals that have celiac disease, for example, with gluten intolerance. There are people in this world that have lectin issues, but I think the vast majority of us don't. And I want to be really careful when you try to cut out vegetables and pastas and beans out of your diet just en masse. You're potentially losing a lot of the really beneficial things that you get from vegetables and from beans and from grains. Yes, people lose weight, but they probably lose weight because they've cut out all of the carbs and all of the breads, not necessarily because they cut out the lectins. Again, I think that there are certain individuals that probably do have lectin sensitivities, but I would be really cautious about any diet that says cut this entire category of foods out, especially things that we know throughout history have been beneficial, like cooked beans, tomatoes. Tomatoes have a lot of antioxidants and lycopene in them that are beneficial. Yes, they also have lectins. So I would say the answer to this question, are lectins bad for me? For the vast majority of us, the answer is no. There's some interesting science that I think bears watching, but again, the vast majority of it is down here in this box, not up here. So we all are always looking for that next thing that makes us slimmer, faster, younger, whatever. And I think the challenge is just cutting out an entire thing in mass is not necessarily the way that we want to do that. Next question. How do steroids affect the gut microbiome? So I did some digging into this, and there clearly are some examples of how steroids impact the gut microbiome, especially steroids like sex hormones. Age-matched, geographically matched, diet-matched men and women have different gut microbiomes. And the data seem to point to the sex hormones having some impact on that. There's some data that talks about that, that certain types of steroids may alter the microbiome, but the data isn't very clear. And again, it still is down there in the association. And one of the challenges with these studies were they are not consistent from study to study. So we can't draw broad conclusions yet. So do steroids affect the gut microbiome? Probably so. In what way? We don't yet have a good answer around that. Does it make a difference to the various organs of the body or the digestive process if the food that we eat is cooked, ground finely, or blended in a smoothie? Or if we want to jump back to I Love Lucy, if it is packaged in Vitamin-to-Vegemin? Does it make a difference? So in some cases, the answer is yes. For example, if you are eating something because of the healthy bacteria that come along with it, the probiotics, cooking it is not going to do you any favors because it will kill all the bacteria. In other ways, if you are juicing, one of the challenges of juicing, and again, hear me, I'm not saying that these are bad. I just want you to be aware of things. When you juice, you're getting an enormous amount of, number one, sugars and a lot of nutrients all in liquid form that rapidly move through and are absorbed by your body very quickly. As opposed to if I eat the orange and I'm also getting a lot of the fiber and the pulp, and that combination is digested more slowly in my system. It's also easy if you're like me, if you have a great glass of delicious tasting juice, I might have a glass of juice that's this big, which is the equivalent of like 4 apples, 2 oranges, a beet, and a rutabaga. Boom, and it's gone. And it's passed through my system, and in an hour, I'm hungry again. So it doesn't seem to matter. Some of the studies that I looked at don't seem to, it doesn't matter if you've shred or ground or pulverized, your body is still getting the nutrients out of those foods. But what is it you are looking to get, and is the process of preparing your meals actually working against you, is an important question to ask. And then I got this great email from a friend who's also in the class that said after the first week, and we talked about your microbiome and about your gut and about testing your feces, this showed up in my Facebook feed. And it's an ad for a company that wants to test your gut microbiome. And he said something to the effect of, should I be afraid? Are Siri and Google and Alexa somehow tracking that I am here and then providing me with this information, knowing that I have somehow, are they looking at my calendar and seeing the word microbiome and doing the search? So this comes to a question that many of you have actually asked me over the last 3 weeks, which is, can I test my gut microbes? How can I test my gut microbes? There are a whole set of different companies that would very happily test your gut microbes. I've got 4 of them on the screen. So from left to right, this is Biotech. So the Greek symbol micro is a U that's got a little funny tail on the front end. So Biotech is a play on microbiome. Thrive, American Gut and one called Biome. And each of these offer different types of testing. So and you can see the price points, and the price points vary dramatically depending on if we're talking about a one-time test or we're talking about a set of 5 tests that you could use over a course of multiple weeks, maybe while you're thinking about changing your diet, or if you would actually like a set of nutritional supplements and probiotics to take once you've had your gut analyzed, or if you'd like a clinical microbiome, which is run in a clinical setting that actually works with a physician on site in one of these organizations working with you and your physician. So that sidesteps some of the direct-to-consumer testing. Or if you'd like a whole set of dietary plans based on what you find. So lots of different groups, including American Gut, which is a citizen science project. It actually started with a GoFundMe page and became incredibly successful. It's done by a group of academicians that gives you information that is very clearly research grade. So yes, there are lots of ways that you can have your gut microbiome tested. I want you to take that with a jar of salt. Well, maybe don't take the jar, but then we have high blood pressure issues and definitely altering your microbiome. But I want you to be aware, based on what we've talked about over the last 3 weeks, there are multiple different types of tests. Am I looking at the whole genome sequencing, the metagenome? Am I just looking at the 16S ribosomal? Am I looking at this from fecal material? Am I looking at this that I've analyzed in a very specific way? What level of detail can I get down to in the categorization? What does that mean? How closely am I able to track this with my diet and my health history and other pieces? So I truly believe that we are reaching a point where this will become an important part of our clinical plan. We will get a sense of the health of our gut microbiome. Are we there? With a few rare exceptions, I think right now this is still mostly just, I'm kind of intrigued by this. This is kind of, I'm kind of interested in what I want to know, but I wouldn't necessarily make lots and lots of clinical decisions based on that information. So if you'd like to have your gut microbiome tested, there are lots of other groups out here. I am not endorsing any of these. I am not paid by any of these. But there are lots of places that you could go if you wanted to test your gut microbiome. Hopefully you are intrigued by what we've talked about, but you also recognize there are a whole lot of caveats before you just suddenly take this and start making major clinical decisions. All right, now let's see if there are questions either from our Jackson Center audience or questions that we would like to have our online viewers that would like to bring up as well. So, questions? Yes? Ooh, how does glyphosate affect your microbiome? So glyphosate is Roundup. It's the weed killer Roundup. Have I seen any papers talking about the impact of glyphosate? There's nothing that comes to mind. I would imagine, all right, so now I'm going to step to the very edge of my knowledge, and I'll look at this week. But I would imagine that any residual glyphosate, and residual, we can have a conversation about how much that actually would be that you're even ingesting, and if it breaks down before it gets to you, but would likely be broken down by the acids in your stomach. So only fragments of chemicals would actually even get to your gut microbiome, and not in its active glyphosate form. I know glyphosate has a really short half-life as well, so it breaks down into inactive byproducts. But again, I am past the edge of my knowledge. So let me dig deeper into that, and I'll come back next week with that answer, or at least with what I can find if I can find anything on it. Other questions? Yes?
I think you said last week that celery, most people don't have the bacteria to the good bacteria they need to digest it.
So for celery, most people don't have the genes in their own intestinal cells to digest it, but there are lots of gut bacteria that are very happy to munch on those polysaccharides in celery. Others. Really? I know I have not answered all of your questions about the microbiome. All right, let me see if we've got any from our online audience. No. All right, well, we'll check back with that a little bit. And I'm sure as we go through this, we'll have some other questions as well. Okay, so this week, I told you the first week that we were going to do this. We were going to speed skate our way through some of our sessions instead of stopping and dropping a line and really digging deep. This is a week where I wish we could drop a line. We could make an entire 4-week series based on the things that we're going to talk about tonight. But sadly, I am going to touch on them in only the lightest way, and I will apologize up front for the reality of that. We're going to start with cancer. This is a part of our microbiome studies that when we first did this 5 years ago, there was almost no information on the relationship between the gut microbiome and cancer, either cancer of the digestive system, especially colorectal cancer, or any distant cancers. This is a field that has really grown in content over the last 5 years. We're going to start specifically talking about the relationship between the food you eat, the way it is metabolized in your gut, especially in your large intestine, and associations with colorectal cancer. So, there are increased risks of colorectal cancer from diets that are high in red and processed meats and alcohol, especially in red and processed meats and in charred meats, meats that are cooked with a heavy level of char, black, thick, almost charcoal-like on them. There may be bacteria in your gut that break red meat into TMAO, trimethylnitroamine ox. TMAO, let's just leave it there. And TMAO has been associated with increased risks of colorectal cancer from a number of different pathways, including as an inflammatory, a pro-inflammatory, an irritant that is increasing inflammation in your colon. And if you remember from our Biotech series a couple of years ago on cancer, inflammation is one of the key drivers of cancer. Continual low levels of inflammation creates an environment that is receptive for some of the other pathways to drive cancer forward. On the flip side, there are decreased risks of colorectal cancer from fiber, and you'll remember we talked about this a lot when we talked about dietary issues last week, and marine omega-3 fatty acids. So these are the fatty acids, the omega-3 fatty acids that you get especially from fish and shellfish, not necessarily the omega-3 fatty acids from plants. The fish, marine ones, seem to be the protective factor. We don't have a good sense of why the omega-3 fatty acids are the protective factor. The fiber, we seem to have some insight. If you remember last week, we talked about the fiber, the bacteria in your gut break fiber down into some intermediates that then are converted further by bacteria into short-chain fatty acids, SFCAs. And one of the SFCAs that we talked about was butyrate. And butyrate is one of the primary energy sources for the cells of your colon. And butyrate also seems to have an anti-cancer effect. So the more fiber in your diet, the more butyrate you are producing, the better the health of your colon cells. So we used to think the relationship between increased fiber and reduced colon cancer was that the fiber essentially allowed food to move through your digestive system faster. It kept you regular, so toxins didn't sit in your gut for long periods of time. That may still be the case, but we think one of the key anti-cancer factors for fiber really is its conversion to butyrate, and that butyrate on its own has this anti-cancer effect. So there are some early studies being done looking at supplementing, adding butyrate into patients' diet to see if additional butyrate on its own, not even the fiber that you convert to butyrate, may actually help reduce risks of cancer. So stay tuned on that. But again, it is another reason why fiber is so beneficial to your gut, because it feeds the bacteria. It is a prebiotic. It provides nutrients for the good bacteria, and it is broken down into butyrate and other short-chain fatty acids that are valuable. And those short-chain fatty acids don't just go to your colon, they go all throughout your body, including to your brain, your heart, your liver, and your lungs. This is a really busy slide. And I'm going to hit the key point that I want to make about this slide is twofold. So this box here, you might be able to read, these are specific strains of bacteria that have been associated with anti-tumor effects. So these seem to reduce the incidence or the frequency or the size of tumors in laboratory studies. The clinical studies are still in process, but there seems to be a set of bacteria that have anti-tumor, or they are protective against cancer. And you can see, maybe if you squint, you can see some of the things. Apoptosis, that means cell death triggered in cancer cells. Anti-cancer immune response activation. That's a fancy way of saying it seems to awaken your immune system to attack the cancer cells. So these bacteria, in some way, shape, fashion, or form, seem to be beneficial in battling tumors, reducing tumors. On the flip side, this sadly larger set of bacterial species seems to have pro-tumor effects. And these specific effects really vary. So this strand of E. coli introduces double-strand breaks in your DNA. It breaks apart your DNA, and DNA breakage is one of the things that can lead to mutations that drive cancer forward. This one actually degrades a molecule called P53. P53 is the guardian of your genome. It is like the traffic cop that makes sure that everything keeps moving, and if there's damage, halts everything. If you don't have P53, you increase the likelihood that damage to your DNA and cancer slips by unnoticed. We won't dig into all of these. These are different pathways that we know are involved in telling cells, hey, let's grow, let's divide, let's ignore the stop signals, let's go crazy, let's party. Well, maybe not let's party, but. And then over here, estrogen receptor activation. So this set of bacteria mimics the effects of estrogen as if you had higher amounts of estrogen. And so estrogen responsive, especially estrogen responsive breast cancers, these cancers respond to estrogen. Estrogen makes them grow. These bacteria mimic the effect of estrogen. We are no longer simply talking about colon cancer here. We're talking about multiple different types of cancer and the impact of these bacteria that live in your gut. Now, let me be careful to say, again, if you decide to go have your microbiome tested and you discover that you have some of these, don't panic, because, for example, this Bacteroides fragilis also has other beneficial value as well. It does things that are positive for your body. And there are some specific strains of bacteria that are good for you, and slightly different strains of the same species have a different genetic recipe enough and are bad for you. So again, we haven't gotten all the way down to this is the good strains that you want and these are the bad strains. And remember, much of this lives in this range. Maybe we'll move a little bit up here, but not all the way up here. But the insight into the role of your microbiome in creating an environment that either minimizes your effects of cancer or increases the environment, puts you in a pro-cancer environment. We are just now, again, beginning to get a better handle on that. And what is the main driver of which of these you have? The food you put in your mouth. I'm jumping ahead a little bit to next week because I don't have enough time next week to dig all the way into all of our pre and probiotics, so I want to give you a preview. We're actually up here in the set of things that I'm showing you on this slide. We've actually done the replication work, we understand some of the function, we've done this in model systems, and we're now moving into clinical trials that in individuals with certain forms of cancer, certain probiotics, beneficial bacteria, may actually be helpful as they are going through their cancer therapy. So many cancer therapies do a number on your gut. They do a number on your digestive system. And clinical trials have shown that certain strains of probiotics, healthy bacteria, actually can minimize some of the upset, the digestive upset and the diarrhea that accompanies many forms of chemotherapy and other forms of cancer treatment. So the clinical tests have shown that. Clinical tests have also shown that there are certain chemotherapies and this new category of immunotherapies that are a really hot category right now. Your gut bacteria, certain strains of bacteria, may inactivate those medications or may actually turn up the activity, creating a toxic environment. So too much of the medicine actually can be even more damaging. It doesn't just destroy the cancer, but it does damage to the rest of your body. So I think we're seeing clinical trials to try to understand what gut bacteria can predict how well chemotherapy and some of these immunotherapies are going to play a role. So already, before we step into many of these immunotherapies, we do genetic testing to see if your tumor has the specific genetic variants that these therapies will be able to respond to. I think we're also going to begin to see some microbiome testing or just some probiotics that may help impact that as well. So this is an area that is really beginning to grow and take root. And I think we're going to start seeing it increasingly in clinical practice. It is one of the places where clearly probiotics makes a difference. I'm not going to be able to tell you a whole lot of other places next week. We'll hit a few of them, spoiler alert, but this is one of those. All right, we're going to dig into some deep science. And before we do that, question from our online audience. Ooh, what is the current most negative thing to the average person's microbiome? I'm guessing that our online audience, that the person who asked this is saying, what do I see as the most harmful thing or the biggest threat to a healthy microbiome? And that's easy. It is our Western diet. It is the huge amount of processed, high caloric, high fat, low fiber, high sugar food that we put in our mouth. And not eating a lot of grains and vegetables and fish and minimally processed food. So I think if there is one thing that we know leads to altered gut microbiome, it is our diet. That would be the number one thing. And I'm guilty of that. I like my fast food. I like my pizza. I like my ice cream. I am trying to not eliminate those, but to bring those in moderation and bring in more whole foods and more vegetables and fruit and minimally processed foods. So that would be the number one thing that I would see as a threat to your microbiome and mine. And at this point, if you are really angry at me, I'm okay with that. There are very few times that I will tell you Dr. Lamb says. Those of you that have been with me year after year know that. This is a Dr. Lamb says. Minimize your Western diet. Trim back all of the fast food and heavy processed, high caloric, high sugar, low fiber foods. And yes, you can take Metamucil, but getting the fiber through your diet instead of through something that you drink would be even better for you. All right, let's jump into some science, as if we weren't doing science already. This may seem counterintuitive, but exposure to germs as a child actually seems to be beneficial and helpful. And our current focus on doing everything we can to create an antibacterial, sterile environment for our children is potentially backfiring on us in pretty significant ways. And the data around this really started in the late 80s with findings that hay fever and eczema, which are both autoimmune and allergy related, seem to be less common in families that had more kids and families that lived on farms and grew up around animals. And as researchers dug into this more, they also found that these immunological disorders, these autoimmune disorders, allergies, eczema, were less common in less developed parts of the world. So in our first world settings, our modern hygiene settings, these incidences were much higher than they were in less developed parts of the world. And I think you can actually see that in this set of graphs. So the graph on the left shows you the incidence of lots of different infectious diseases and how they have dropped over the last 50, 75 years. And on the right, the graph shows you a number of these autoimmune diseases and how their incidence have gone up. And so this led to this concept, which was originally called the Hygiene Hypothesis. Now that name's changed, but I want you to understand the concept Hygiene Hypothesis, which suggested that the industrial way that we've lived, living in highly sterile societies and vaccinating against disorders, certainly reduced infections and death from these kinds of pathogens and infectious diseases, but might have led to an unintended rise in autoimmune and allergies. And before you walk out of here saying Dr. Lamb is anti-vaccine, that is not what I'm saying. That is not what I'm saying at all. I'm saying that in a world that has minimized bacteria from a number of different ways, we may have unintentionally gotten rid of bacteria that actually have been beneficial to how our body figures out how its immune system needs to work. That's where I'm going with this. The problem with this concept of hygiene hypothesis was the public, the media talked about this as the too clean syndrome. So we are too clean. And that put a whole focus on something completely different. It implied that there was a whole storyline that arose around people being too clean and that people were being made to feel guilty for living in clean environments. So the concept was changed, the hypothesis was changed to something called old friends hypothesis. And the old friends hypothesis says that humans for thousands of years, tens of thousands of years, lived in an environment that had lots and lots of bacteria. We lived not on sterile floors, we lived on the dirt. We lived very close to outside. We lived outside, or we lived in homes that were not airtight with lots of interaction back and forth. We spent a lot of time outside. We had very few individuals, if any, that were born by cesarean section in a hospital setting. Our water was not necessarily sterilized and chlorinated. Our food was not always cooked and processed in a way that got rid of and killed bacteria. And that when we lived in those conditions, everyday bacteria just became part of our community. It was just part of what we grew up in. It was in our mouth, it was in our gut, it was on our skin. And these old friends are things that our modern way of living have shifted to the side or have minimized our exposure and our impact to. And so children that are born today, their early years of life are in a setting that are much more sterile than individuals that were born 100 years ago and what their early years looked like. So that's the concept of the old friends hypothesis. And it's gained a significant amount of traction because we've shown it in research studies with other animals. We've shown it in some clinical work. So this pretty significantly seems to be the prevailing concept, that the loss of these old friends may inadvertently increase our disease risk. So how? All right, this is about the most scientifically detailed that we're about to get. So those of you that find yourselves feeling like you are back in high school or college chemistry, and that makes you uneasy, if you would like to just close your eyes and think of something lovely like cake or Disney World or whatever, you do so. We won't go into these slides for long, but here's the detail piece around that. So your gut is clearly a digestive organ. I think we've explained that in detail. But it also is an important part of your immune system. And so you have all throughout your gut, you have multiple different cells, your own cells and your microbes that are helping sense what's going on in your gut and passing that information to your immune system. One of the examples of that is a part of your gut, a type of your gut called a Peyers patch. And they are these, you can see them on the slide on the screen behind me. There are lots of different cells involved in the immune system that are in these Peyers patch cells, including cells that actually, it's hard to see, cells that actually send up projections through your gut epithelial cells up into the mucus. So here's the mucus layer, and then above it is all the food that's passing through your large intestine. So these cells send up little antennae, and they can sample what is present in your gut. So it's a way of saying, hey, this bacteria is here, or hey, I'm seeing this protein, or I'm seeing this unusual thing that I haven't seen before. And then that information is relayed back to other parts of your body, and including to your immune system. So there's this constant monitoring back and forth. It's fascinating. There are always these reconnaissance missions saying what's present out there, how much is out there, and then sending that information to your brain and sending that information to other parts of your body and to your immune cells. The thinking now, the science suggests that this kind of routine interaction in an infant's gut is really important because it helps an infant's immune system begin to calibrate. It helps the immune system recognize what is myself, what are my own cells, what are the bacteria that are just kind of present, hanging out with me at low levels all the time, and what is really worthy of, this is unusual, I need to mount an immune response against this. So it's that concept of myself, my friends, and my foes. And that's this interaction. And the thinking is that in infants that don't have a lot of old friends, a lot of those regular commensal bacteria that hang out in your system, it may make it really difficult to regulate, to know when I should not respond, when I should respond, and when I should just hold back and not mount an immune response. And it is looking like this takes place through a set of proteins called Toll-like receptors. And Toll-like receptors receive information from your gut, from your microbes. They use that information to then pass signals to your immune systems and to your immune cells. And they actually, so your immune cells receive this information, they pass information to other parts of your immune cells that say, hey, only above a certain threshold of these unusual things should you now mount a big immune response. And below that threshold, this is the stuff that we see every day. And this is your own cells. You don't need to respond to your own cells. Your immune system goes through this training to be able to say, well, that's a protein I haven't seen before. Is that from my own body and I need to ignore it? Or is that from some foreign body and I need to attack it? And if it doesn't have that training, it will say, that's a protein I haven't seen before. We're going to attack it, even if it happens to be your own system. And autoimmune disorders arise because your body fails to recognize your own cells and mounts an immune response to damage and destroy those cells. And the thinking is that if in pre-industrial times, and when we had dirt on the floor and we had some bacteria in our water and in our food, we were able to train the immune system when we were young. And that potentially we've lost the ability to train very well, and now we are seeing this rise in many of these autoimmune disorders. Does that make sense? We covered a lot. That's a lot of deep ground and Toll-like receptors and all those pieces, but that's the concept of the old friends hypothesis, and it has gained a lot of research and clinical support. I think this is likely very, very valid. So the question now becomes, if this is what's going on, and if today this is where we are, how do we figure out how to send signals through our microbiome to our developing immune system to train it or to retrain it appropriately? Can we retrain it, or is there a window only in infancy and early childhood where you have that ability? And that's a question that's still up in the air right now. So there are multiple autoimmune disorders that have been associated, bottom box, with alterations in your gut. And you see those on the screen behind me. And they range from thyroid-related immune diseases like Graves and Hashimoto, and skin-related autoimmune diseases like eczema, and type 1 diabetes, and systemic autoimmune like lupus, and then central nervous system related disorders and neurodegenerative. And we're going to touch on just a few of these. I know it's almost impossible to read the words on that slide, and I apologize for that. This is a figure that came out of this research paper, but one of the things that I want you to notice, increased intestinal permeability. That's here, that's leaky gut that we talked about last week. It's here, it is here. So those are common features. You may also see reduced Akkermansia, Akkermansia mucinophilia. That's one of the bacterial strains that we talked about last week, associated with reductions in that was associated with obesity, and reductions were associated with type 2 diabetes. So you're going to see some of the same players even as we move our way, as we try to get our way further up this set of research. I love this quote. You do not have this quote in your handout, but I just want you to take a look at this. I'm going to actually read this because I think this is, well, first of all, I think it's very telling. This is by Jane Danska, who studies type 1 diabetes at the University of Toronto and Hospital for Sick Children. There's been a lot of Kool-Aid consumed, including by us, about the role of the gut microbiome in fill-in-the-blank autoimmune disease. So let's just pause right there. There's a lot of this going on, and a lot of people get really excited, including researchers, about the relationship between the gut and autoimmune diseases in general. One of the mechanistic challenges to researchers is how do you connect bacteria that are growing inside the tube of the gut with immune responses against completely different tissues? So I got a different gut microbiome, but what does that have to do with my immune system in my pancreas or on my skin? What's the linkage there? And that really is the missing link. So, because you could simply have an altered gut microbiome because you've now got this disease. It could be a consequence of, not a cause of. So how do we begin to get at an understanding of that? Yes?
So I think I read that 80% of your immune system is in your digestive system.
Ah.
Where's the 20%?
So you read that 80% of your immune system is in your digestive system. Where's the other 20%? Those numbers, that 80-20 ratio, really depends on what you're measuring. But there is a significant part of your immune system in your gut based on the slides that we just went through. Much of the rest of that, your immune cells, the cells of your immune system are birthed in your bone marrow and travel to your thymus where they're trained, and then travel throughout. So you've really got them circulating throughout your body, but the birth of those is in bone marrow. So this question, what's the connection between the two? Well, we got a glimpse of that really just a few weeks ago with a paper that came out by this author, by Dr. Danska, looking at gut microbiome and type 1 diabetes. And I'm going to walk us through this, and then we're going to take our break. Type 1 diabetes, historically called juvenile or insulin-dependent diabetes, although not everyone who gets type 1 diabetes is a child, is an autoimmune disease. It occurs because your body attacks the cells in your pancreas that produce insulin. And so you now lose the ability to produce insulin. You have to take it through a syringe. The increase in type 1 diabetes around the world, there have been enormous increases over the last 20 or 30 years without a clear understanding of why, what is driving this increase, just like we see increases in lots of other autoimmune disorders as well. We've begun to get a sense we can look for what are called islet autoantibodies. Before an individual develops type 1 diabetes, you can look at their blood and you can look for antibodies that specifically are designed to attack proteins on the islet cells of your pancreas. So you can begin to see a person's immune system turn against their pancreas and start saying, oh, that protein, that's a foe, I need to attack it. Oh, that protein, that's a foe, I need to attack that. So you can watch as children accumulate autoantibodies against their islet cells, and then at some threshold, and it varies from person to person, they cross into type 1 diabetes and symptoms. So you can measure an individual's disease progression, not always, but sometimes by the onset of symptoms, but you also can look earlier at the development or the progression of these autoislet antibodies. That's the background. Now here's the research. It's really elegant research, but also kind of complicated. These researchers looked at bacteria that are commonly found in a typical or a healthy gut, and they created a gathering of them. They grew a gathering of them, so I guess they kind of made and they didn't capture all of them. I think they captured like the 50 maybe, that number's probably wrong, but some dozen of the common bacteria found in gut, and they grew them in the lab. And then they took blood from patients with type 1 diabetes, Crohn's disease, and patients without diabetes. They took that blood and they took the serum from that blood and they added it to these bacteria. Because the question is, did the bloodstream of these patients actually have antibodies that were attacking and killing typical bacteria in the gut? Because they know that the microbiome of patients with type 1 diabetes is different than the microbiome of patients without. And what they found was that patients with Crohn's disease and patients with type 1 diabetes had antibodies in their blood against typical bacteria in their gut. So they were attacking and destroying the normal flora and fauna of bacteria in the gut. Patients that didn't have Crohn's disease or type 1 diabetes didn't have these antibodies. So this image, so here are the antibodies, so anti-commensal or typical bacteria antibodies. So these individuals that had type 1 diabetes had these antibodies in their blood attacking the bacteria in their gut. They then said, okay, well, if individuals with type 1 diabetes have these antibodies, do individuals that have the autoantibodies that are a few steps before the onset of symptoms, do they also have the gut antibodies? And the answer is yes, they do as well. So before they ever have symptoms of type 1 diabetes, they seem to be attacking the gut microbes at the same time that they're beginning to turn against the cells in their pancreas. So there's a link with the immune system attacking the gut microbes and beginning to attack the pancreas. What that connection is not understood. But it is the first hint of something that links an immune response to the gut with an immune response to the tissue that's actually causing the disease. So the work of these researchers now is to see if this presence of these antibodies in the bloodstream can potentially be a biomarker that's present even before they begin to show antibodies for their pancreas. In other words, is this an even earlier biomarker for the onset of type 1 diabetes? And can you catch it early enough before you begin attacking the pancreas that you could start thinking about doing something to prevent the pancreas from being attacked? It's a lot of details all in that one figure. And again, that's one paper in a relatively small number of kids. It has not yet been replicated, but it's really elegant data that now has a lot of people talking because it potentially gives some sort of functional mechanism around how your autoimmune disorder could lead to a set of symptoms and an altered gut. It isn't saying that the altered gut is causing the type 1 diabetes. It's saying that somehow the body is attacking itself in the gut and in the pancreas, and maybe the gut is the first place that it attacks. So can we figure out why it's attacking the bacteria in the gut? Okay, we're going to take a break. We are going to take a 15-minute break, and then we'll come back and we'll move our way through the rest of our talk. All right, I have 2 questions from our online audience, and I'll take them as everybody's kind of gathering their seats. The first one is that there's a lot of popularity around apple cider vinegar right now as a dietary supplement, and is there any evidence that suggests that there's an effect of apple cider vinegar on the microbiome? And none of what I've seen, I actually looked into this about a week and a half ago. I have not seen any evidence that apple cider vinegar impacts your microbiome. Second question. Children often drop food on the ground and then still eat it, i.e. the 5-second rule. I would imagine some of us in this room might also still follow the 5-second rule. Could this potentially be strengthening their immune system? I think the first question is it depends on what they drop it into. But I think that eating food that's dropped on the... Wow, how am I going to answer this question and not get... Yes, possibly they might be strengthening their immune system. I think there's nothing wrong with eating some dirt now and then. But again, it depends on what you drop it into. Let's be really clear about that. If I drop my jam sandwich jam side down in a cow pasture, I'm probably not going to eat it no matter how few seconds it lands on the ground. But all right. Okay, I want to take us into our final section, which is the gut-brain relationship, the relationship between your microbes and your brain. But I want to start by telling you this really bizarre story, again from Ed Yong, who we've talked about each of the last weeks. This is a story that he wrote at The Atlantic, and you can find it just by typing in Ed Yong and zombie ant fungus. Okay, so this is a fungus, and this fungus infects lots of different insects, but this particular one infects carpenter ants. And when it infects a carpenter ant, the carpenter ant begins to behave in very strange ways. It begins doing multiple grooming, and then it ultimately climbs its way up the stalk of a nearby plant, and it climbs to about 25 centimeters, a very precise height. And then its mandibles, its jaws, clamp on to the stalk, and it's actually called a death grip, and it just stays there. And what's happened is the fungus has essentially, as it's grown throughout the ant's body, it has taken over the ant's body. It has created a buffer between the ant's brain and the ant's body and the ant's movements, and the fungus now controls the movement of the ants. The brain is still functioning, but the fungus is at the steering wheel. And so the ant clamps on this death grip on the stalk, And there it stays, and ultimately it dies. But as it is dying, the fungus grows and sends a stalk out through the back of its head. It takes about 3 weeks. There's the stalk. You can see its jaws are clamped onto this. There it is. And then there's a bulb at the end. This is afterwards. But the bulb is full of little fungi, little spores, and it bursts open and the spores rain down below. And usually, because the carpenter ant has climbed up a stalk as it was following one of the ant trails or near its colony, the spores rain down on the ant's co-workers, turning them into zombies as well. Yeah. Yeah, there's actually a game that uses this storyline. There's a popular book series that uses this zombie storyline. But this is an incredible example of microbial behavior modification. The microbes, the fungi, are controlling the behavior of the ant. And that's a great segue as we talk about are microbes potentially controlling parts of our behavior. Clearly not like that. But the concept, so this leads us to this idea of what's called the gut-brain axis. And the gut-brain axis says that your brain controls actions of your gut. It regulates your gut. It regulates how your gut contracts and relaxes and how food moves through your digestive system, and things like the flight or fright response, that those signals, those hormones from your brain that regulate your gut. But it also says that your gut is sending information back to your brain and impacting your brain, some of which through the microbes in your gut, some of which is just through the cells that line your intestine. So this happens in a number of different ways. Some of it is through your vagal nerve. The vagal nerve runs from your brain down through your body, and it innervates your gut. It innervates other parts of your body as well. But it is one of the ways that your brain receives signals. So signals about your appetite. I'm hungry, I'm full. Some of those signals move through the vagal nerve back up to your brain. But other signals may move through your bloodstream back to your brain and cross the blood-brain barrier in some cases. So the brain-gut axis suggests that your brain is controlling and sending signals to your gut, and your gut, and therefore the microbes in your gut, are also sending signals back to your brain. Your gut is one of the largest producers of neurotransmitters. Your gut, in some cases, produces more neurotransmitters than your brain does. Most of your body's serotonin, which is a mood regulator and involved in sleep and in emotion, most of that is actually crafted or designed. Most of that comes from your gut, not from your brain. And so what I've got on the screen, on the slide, are a whole set of different bacterial strains and the neurotransmitters that they create. And you see serotonin in almost every single one of them. These bacteria are producing lots of neurotransmitters. The question is, how many of these neurotransmitters actually make their way back to your brain or to other tissues? And so in some cases, these pass through your gut, through your barrier, but they don't get into the blood-brain barrier. In some cases, they signal the vagus nerve, and then that signals in your brain. In other cases, it appears that there are precursor neurotransmitters that can cross the blood-brain barrier where they're converted into active neurotransmitters. So the bacteria in your gut and the cells in your gut are producing signals that actually are impacting your mood, your behavior, and your sense of emotion. So not nearly as dramatic as what we just saw with the zombie ant fungus, but your microbes are, at least there's significant evidence, are capable of shifting and altering your behavior. So this concept of a gut feeling actually has some scientific validity to it. Neuropsychiatric disorders, a number of them, the ones on the screen behind me, have all been associated with altered gut microbiomes. So differences in the kinds and populations and frequencies of bacteria in your gut. Now you notice I said association. Remember I told you at week 1 we were going to spend a lot of time in this box. You are sick of this box. You are ready for me to get all the way up here. And I keep telling you it's down here. Lots of them. Depression, schizophrenia, bipolar, autism spectrum disorders, ADHD, Parkinson's, and Alzheimer's all show altered gut. But again, it's an association. In many cases, it has made its way up to replication. I'm going to show you a few examples that have moved their way a little higher up the chain, but most of it is still down here, and we haven't figured out if it is cause or effect. We're going to pick 3 stories. We're going to talk about depression, autism spectrum disorders, and Alzheimer's disease. And I'm going to touch on these very lightly, and I apologize that I am not going to be able to dig deeper into these. The first one is actually research. For a long time, there have been suggestions from small patient studies and from mouse work that your altered gut could play a role in the onset and the severity of symptoms of depression. But there wasn't a lot of human clinical data until studies that were published literally 2 weeks ago. Yeah. Every single week that we've been here, there's at least one story that I'm showing you from a paper that has come out since we started this series. That's how quickly the science continues to roll out. So this is a study that was done looking at Belgians. This was actually meant to be a study of the healthy microbiome of over 1,000 Belgians. And so they sampled their gut microbiome, and they asked a whole lot of questions about the individuals, and they realized that there were about 173 individuals out of this 1,054 Belgians that said, I have been diagnosed with depression, or all of these individuals also filled out a quality of life survey, and their scores on the quality of life survey were really poor. And poor quality of life surveys, scores on this particular survey are associated with depression. So they looked at this 173 individuals, and they looked deep into the microbes that they found in their gut. And they found that there were 2 specific strains, 2 specific species of bacteria that were present at really, really low levels, that were present at high levels in all of the rest of the non-depressed Belgian population. And here are those 2 types of microbes that were missing. They then, remember I told you the key is can we replicate it? They then went to over 1,000 Dutch individuals. And they looked at the same, they looked at a subset, about 175, 200 individuals who also had been diagnosed with depression or had low scores, and a set of patients that had been diagnosed, that had a clinical diagnosis depression that they also added in. And in those patients, the Dutch patients, they also were missing the same set of microbes. And the specific set of microbes that they were missing seemed to do one of 2 things. They normally produce a significant amount of dopamine, which among other things contributes to mood, or they normally produced a lot of butyrate, which is a short-chain fatty acid that we've already talked about, that we know butyrate travels to the cell, to the brain, and is important in brain health. So they've at least got some functional suggestions. So this one paper gives us this, and some early indication here that maybe there's something about these specific strains that when we don't have them in our gut, they are altering our mood by altering the responses in our brain. So this group of scientists is actually now doing mouse work, trying to look at mouse models of depression and see what happens when you alter mouse microbiome. And they actually, another researcher, not part of this study, has started a clinical trial looking at patients and trying to, either through something we'll talk about next week, fecal microbiome transplants or probiotics, trying to boost the levels of these two types of microbes and see if we see a difference in depression scores among patients. Depression is one of the knottiest, K-N-O-T-T, not naughty as in bad, but thorniest issues for us to try to get a handle on because it seems to be so complex. If in fact a portion of that resides in our gut, who would have thought the bacteria in your digestive system could be influencing your mood in such a strong way? So stay tuned for this study. Gary.
Did they have any, maybe I missed it, but did they have any information on the diet of the people 173 years?
Oh, that's a great question. Did they have any information on the diet of these individuals? Did we know what their diet looked like? Could their diet be driving their microbiome? I think because this study was specifically looking at the impact of normal microbiome, if I remember correctly, they were actually able to control for variations in diet, and they were able to control for variations in depression medication. Because you might also expect certain depression medications may have an impact on the bacteria that are present. So if I remember correctly, the answer to that question is yes. All right, now let's talk about autism spectrum disorders. And let me tread into this really, really carefully, because this is very preliminary data. This is not go out and tell your friends and tell your acquaintances who they themselves or someone in their family has autism spectrum disorder that they need to go find some probiotics. Let me be really, really clear about this. But there is an increasing number, a lot of evidence that is now suggesting that at least a subset of individuals with autism, that are on the autism spectrum, have pretty significant gut issues as well. Not all patients with autism do, but a number of them do, and the ones that do tend to have more severe symptoms, in part probably because of all the issues associated with the discomfort around their gut. Researchers have identified and have replicated that there are differences in the bacterial population in the gut of these individuals, or at least a subset of individuals with autism spectrum disorders. And they also are finding differences in the regulation and the signaling of their immune system, which takes us back to some of the things we talked about a little bit earlier tonight, that alterations in the gut may be also leading to alterations in the immune system, and together that along with genetic factors, because autism is a complicated issue. It has genetic components, it has environmental components, and it looks like some of those environmental components in some individuals may actually be also associated with their microbiome. There was a study that was done a few years ago looking at a mouse model of autism. And let me say right off the bat, it was a mouse model of autism that was created in a chemical way that does not mimic what we understand about autism, but the mice have very similar sets of autism symptoms. So it is the model that we work with. So there's a big caveat around this mouse model. But they found that when they gave the mice a very specific probiotic, that the mouse's repetitive motions, the mice buried marbles in their bedding, and they buried them over and over and over, and there were some vocalization differences in the sounds that they made, that when they gave the mice these probiotics, those behavioral responses significantly diminished. Yeah. Now again, studies in mice and studies in people are not the same, and often things that we see in mice are not what we see in humans when we take it to that. So I want to be really careful. Again, I'm not telling you that you need to go put people on probiotics, because a lot of people read those studies and went and put their kids on probiotics. The clinical trials around that have yet to still be, I think they've been started, but have yet to be done. But there are some growing indications that with at least a subset of patients that are on the autism spectrum, that a portion of their symptoms may be due to gut dysbiosis, to altered gut issues. And then as we wrap up, and then we'll have a little bit of time for questions actually, in Alzheimer's disease, and this and Parkinson's is way down here. But there seems to be alterations in the gut microbiome of individuals with autism. Now, whether that is early and is a causative issue, or whether that is later on is not yet determined. But there seems to be an increasing theory around autism around inflammation and low-level inflammation. And many of the things that we've talked about the last 3 weeks with alterations in your gut microbiome lead to low-grade levels of inflammation. So this is an early finding without a lot of data behind it, and certainly not a lot of functional data. But it's going to be interesting to see if that's a driver of symptoms or if that's something that happens as individuals move into the other symptoms of Alzheimer's. Along those same lines, though, there was a paper that was published earlier this year also about a relationship between a microbe, this time in your gums, and Alzheimer's. And it created an awful lot of media and a lot of stir. Many of you know that about every 2 weeks I publish a blog series. It's called Shareable Science. Last Monday, we put this story up on Shareable Science that talks about the relationship between the gum microbe. It's actually one of the gum microbes that causes chronic periodontitis and chronic gingivitis. And why does that gum microbiome seem to have some relationship to Alzheimer's? And it has to do with, periodically, the act of brushing, flossing, going to the dentist, or just chewing. So I did not just give you an excuse to not brush, floss, or go to the dentist, unless you're just going to stop chewing, which means you can't have cookies anymore. But periodically, this microbe that lives on your gums gets into your blood system. And there's some evidence that it can travel to your brain. And this is work that was done in some human populations, but more in some mouse studies. So trying to get at some of this information. And the thought, the suggestion, is that if this makes it to your brain, this microbe produces proteins that actually lead to inflammation. And that the inflammation is one of the, as your brain tries to deal with the inflammation, the formation of amyloid plaques may be a part of that. Now, the blog post goes into more detail on this because it takes a traditional way of thinking about Alzheimer's and turns it on its head. So I'm not going to dig deep into it, but I'm going to point you there if you want to learn more about that. Again, this is not saying that we need to quit brushing and flossing and going to the dentist. This is also not saying that all forms of Alzheimer's are caused by gum disease. Let's be really, really clear about that. It is a maybe possible mechanism that we have yet to get all the way up the level of evidence on. But it got a whole lot of press and a whole lot of stories, and we're talking about microbiome, so that's why we decided to make it one of the Shareable Science articles. But you can just go to whatever your favorite search engine is, HudsonAlpha Shareable Science, and you'll get to it about every and if you want, you can get a notice every time we post a new Shareable Science story. It's just a way to keep you informed about some of the things that are coming out in the news in between our Biotech 101 and 201 sessions. So with that, we got a couple of minutes. Let's see if we have any questions from our audience. So Hugh, and then I'll come right back here to White.
Okay, yeah. Now, what is the effect, if any, on the microbiome of the surgeries on the stomach? First, the bypass, gastric bypass, and that has been transcended more recently by veritably removing the
It's a great question. So what's the impact of gastric bypass and the Roux-en-Y bypass, where you essentially take large parts of the stomach out of the way? What's the impact of that on your gut microbiome? It's a pretty significant one. We hit this in detail in one of our previous sessions, so I'm going to refer you back to those handouts. I think it was last week, but the take-home is it changes the makeup of the bacteria in your downstream gut, which is not surprising because you're changing what your large intestine is seeing. It's now seeing a lot of food that traditionally would have been digested earlier up in your stomach and small intestine. So the data suggests that those kinds of bypasses make major changes to your gut microbiome and may be part of the reason why you begin to see improvement in things like insulin resistance before you even begin to lose large amounts of weight from the bypass itself. So yeah, exactly the long-term impacts of that, not yet fully understood.
Going back to the slide that was about gut microbes and type 1 diabetes, what is HLA and DR3 and DR4?
I should have talked about that. I apologize. Thank you. That's kind of important. So going back to the type 1 diabetes image, which is right here, I left out a really important part of this story because I was thinking about break time. I told you that these patients showed that their blood produced antibodies against their gut and against their pancreas. But the key piece I left out is that in the human leukocyte antibody gene, HLA gene, so it's a gene that plays a major role in how your immune system responds, the individuals that had these two specific genetic variants, these were the individuals that seemed to have the biggest impact on attacking their bacteria. And we knew from previous studies that DR3 and DR4 at the HLA gene were risk increasing for type 1 diabetes, but we didn't realize that the connection between these high risk factors and the presence of these antibodies against your pancreatic cells and against the bacteria in your gut. Individuals that didn't have these high risk factors let's see if I can get this right, did not seem to have as much. You know what, I actually brought this with me because I was afraid that I would mess this up. So let me just read this. Initially, the authors did not identify different responses to the gut microbes between the children that had these antibodies in their islet cells and controls. But then they sorted subjects by their HLA genetic information, and they found that it was having these antibodies and having these high-risk variants that predicted the development of type 1 diabetes. So it wasn't simply that you had the high-risk genetic factors. It wasn't simply that you had the antibodies. It was the combination of the antibodies and the high-risk genetic factors that were the best predictors of developing type 1 diabetes. The A stands for antibody. So it's human leukocyte antibody, HLA. Why, thank you so much for bringing that up, because I would have left out I did leave out a really important piece, so thank you for bringing that up. We got time for one more question, and Emily, you were the first, you were the lucky one. Woo, how does stress affect your microbiome? In all kinds of ways. Partly through the gut-brain axis, we think. But individuals that experience stress, early life stress, even later in life stress, the thinking is that the stress in the body and the stress in the brain sends signals that actually, number one, alter the way your gut, the way food moves through your gut, the way your gut contracts and relaxes, but also seems to in some unknown way, shifts the balance of what thrives in your gut. So being under stress, immediate high stress or long-term stress seems to shift what thrives in your gut microbes, in your microbiome. We don't really understand the details, but stress and diet, two key things. Quit eating the Western diet and be less stressed. All right, I'll see you guys next week. We will do probiotics, prebiotics, and antibodies. Have a good night, everybody.